Wow. My name is Alessandro Larcher. I am a urologist and I'm responsible of the local VHL program. The inception of our program was 2021, so we are pretty young as a center, and today we have the amazing honor to host you. So on behalf of our community, our patients, our staff, the residents of our program and the students of Salaferi University, thank you very much for being here today. Welcome, welcome, welcome. Let me introduce all the friends and colleagues who worked together with me and VHL Alliance for the realization of the meeting. Ruhi Dir, Oton Ileopoulos, Andrea Salonia. Andrea Salonia is the person who gave birth to our program since we were managing patients without a very structured fashions since more than 20 years, and he basically came out with the idea of create the structure that is our today program. And the meeting and the event would be not possible without the amazing effort of VHL Alliance. So thank you very much, Joshua Mann and Janet Thompson. Thank you very much and have a great meeting.
Thank you. Welcome everyone, thank you for coming. On behalf of the organizing committee and the VHL Alliance, I would like to thank the Italian colleagues for hosting the meeting. Also, I would like to thank very much, we would like as an alliance to thank two families that donated very generously for the meeting and for research support. This is the Becky family and it also the Rath family. They have been very strong supporters of our research efforts in clinic and basic science. So we have a tight program. We try to put together a mixture of clinical and basic science information. I think that you're going to hear over the next 2 days insights in the new developments of basic science and also an update on the clinical capabilities and experience that we have in the VHL. We'll try to keep the schedule as possible. And again, thank you very much for being here, and I hope we all hope that you enjoy the meeting, and we would love to have your feedback. Right, so it's my pleasure to introduce the first speaker, who is going to give a very special lecture. This is Jim Brugarolas from you. Oh.
I think we have Kim before Jim.
What?
Kim Rathmell. The video.
Oh, we do have the video for Kim.
Do have a video,
oh, okay,
yes.
Great. So Kim, through a video, is the director of the NCI, will introduce Jim. Do we?
Hello and welcome. I'm so glad to be able to send a recorded message, even when it wasn't possible to be there in person. I wish I could be there with you. Milan is such a beautiful city, and this meeting is one that I look forward to every year and have attended more years than not. As you know, I'm still actively engaged in the VHL world. In work that was completed as I left Vanderbilt, graduate student, now Dr. Melissa Wolf, showed that the presence of a VHL mutation led to changes in secreted factors that drew a specific subset of macrophages into the tumor. The implications for immune response are clear, influencing how T cells respond and function. A second study, led by Dr. Zach Bacigalupa, examined a metabolic program governing acetate metabolism. He showed that HIF stabilization in a setting of VHL loss, which you all know as the key driving factor of VHL mutation, was still dependent on a metabolic signal. When that signal was blocked, HIF2 dropped right away. This study adds to the growing list of ways that we can target this cancer-causing signal, many of which I'm sure you'll talk about at the meeting. This meeting is a highlight for me and has played a prominent role in so many ways in my career development. I've met students at this meeting who went on to do postdoctoral studies with me and who are now leading their own labs. I built connections, networks, collaborations, and friendships. The model of blending patients, physicians, and researchers into one meeting really makes this event special. This experience has shaped how I think about and run the U.S. National Cancer Institute. One of the things you have done is show how disciplined observations and creation of a dedicated network can make such a difference for one rare disease. You knew that by really understanding the science, there would be a difference. First, there were good diagnostics. Then there were guidelines for intervention, designed with the patient in mind and optimizing care around them. Good surgical approaches, effect-minimizing procedures, and times when we just watched. Then there were groups able to try the first drugs with a glimmer of hope, early studies with VEGFR inhibitors. But most of all, you refused to take no for an answer when someone said HIF-2 was undruggable. It just wasn't druggable with the technologies we had then. The technologies came along, and just like that, HIF-2 inhibitors were here. And then, willing and brave patients stepped up to try this drug. I remember those who came forward in Nashville vividly. And today, we know there is an option to control VHL tumors without surgery. I intend to extend this energy and momentum to cancers across the spectrum. We still have a lot to learn. If there are ways to prevent these tumors from ever forming, what is the optimal time, duration, and setting to use the current drug? How do we assess the next treatments that are now on our doorstep or that will be coming in weeks, months, or years ahead? We still need to use science to understand all the ways that VHL genetic alterations impact a person and develop effective tools to allow a personalized approach to managing an individual's health. You have also taught me how we do that best. Using an all-in approach, engaging patients, families, and caregivers at every step. Having doctors, nurses, scientists, and communities listen to each other. Learning from each other, taking action together. This is a new era for cancer research, and the NCI is there to approach with any means possible. Our disciplined dedication to ending cancer as we know it. Thank you again for being part of this incredible journey. I'm there with you, along with all of the NCI. Have a great meeting. Ciao, arrivederci.
And now I get to introduce Jim. Okay, so Dr. Brugarolas is an established senior investigator that contributed to the field very much. He's a very important figure in the VHL research. He trained in Spain, actually. He finished in medical school in Spain. He went to complete his PhD with Tyler Jacks at MIT, working in animal models for cancer. And then he joined the fellowship at Dana-Farber Harvard Cancer Center, went to the lab and advanced majorly our understanding of VHL basic mechanism, linking mTOR and HIF function. He left Boston and then he established his laboratory at UT, Southwestern, at Dallas. He's currently the leader of the renal program. He's the director of a multidisciplinary SPORE grant for kidney cancer. And he provided seriously, and this is not leap of service, very significant insights in the genomic fabric and the pathways that they evolve after VHL inactivation and sustained renal cell carcinoma. Today he's going to talk to us about a very exciting problem. So you all know we have Belzutifan as a treatment for renal cell carcinoma, and one of the things that we're all looking for is biomarkers and understanding which tumors are sensitive, why they're sensitive, and how they develop primary or secondary resistance to the treatment. So Jim has the latest and the most hot. Please welcome.
Thank you, Othon, for that very kind invitation. Great, thank you. So thank you for the introduction and thank you for the invitation. I'm delighted to be here with you all. So I understand we have a number of students in the audience. How many of you are students? All right. Well, so these stories for you, because I think most of the faculty are probably familiar. I'm going to show you how we went from what Othon and Kim referred to as an undruggable target to a first-in-class drug. And this was done primarily at an academic center, as you will see. So these are my disclosures, and there are some that are relevant because there are patents and applications related to work in HIF2 and Rapalogs. So you're all familiar with this, and this is a very simple schematic. So this is taken from an old editorial, and it shows how the VHL gene, which was discovered back in 1993, encodes a protein that is critical in this process of regulation of hypoxia-inducible factor. You're all familiar with the work of Bill Kellen, Peter Ratcliffe, and Greg Semenza, which led to the Nobel Prize in 2019, focusing on these hypoxia-inducible factor proteins. Now, it's interesting that as important as VHL is for kidney cancer, VHL alone is not sufficient for kidney cancer development. So we showed that back in 2014. We showed that mice that had VHL mutations did not develop tumors, but you needed to mutate VHL with PBRM1 or VHL with BUB1 in order to develop kidney cancer. VHL alone was not sufficient. And as you all know, these genes all reside in the 3' arm, the short arm of chromosome 3, that is frequently deleted in both sporadic as well as familial VHL syndrome. Nevertheless, even though VHL loss is not sufficient, we know that VHL loss results in constitutive activation of HIF, which is likely the main effector downstream. The HIF-alpha, HIF-2-alpha protein specifically, was discovered by Steve McKnight, who you can see here. He was the chair of biochemistry at the time at UT Southwestern Medical Center. The gene is called the EPAS1 gene. And subsequent studies at UT Southwestern by Rick Bruick and Kevin Garner identified a small cavity within the PAS-B domain of HIF-2-alpha. So as many of you know, the HIF-2-alpha protein forms a heterodimer with HIF-1 beta, and it functions as a transcription factor. So it binds DNA, these hypoxia response elements. And you can only bind DNA as a dimer. You need to have both arms of the tweezer if you want to be able to bind DNA. So these investigators performed structural studies of the PAS-B domain, which led to this unusual discovery or this unusual finding of a cavity within the PAS-B domain of HIF-2-alpha. And they hypothesized that this may provide a foothold for a drug. Transcription factors are challenging to drug because they have globular surfaces, but here we had a highly structured cavity. So at UT Southwestern, there was a screen of our library of compounds. It's an academic library that has 200,000 compounds that were tested for the ability to bind the HIF-2-alpha PAS-B domain and trigger the dissociation from the PAS-B domain of HIF-1 beta. And through primary screen and secondary screen and so on, this led to a lead molecule that had a KD of 70 nanomolar. So this set the foundation for what then became the founding of Peloton Therapeutics by Steve McKnight. So this was a biotech company that is a spin-off of UT Southwestern Medical Center that was built to capitalize on the intellectual property of these first-in-class small molecules that bind HIF-2-alpha. So these molecules, what they are expected to do is they are expected to bind HIF-2-alpha and lead to the dissociation of HIF-2-alpha from HIF-1 beta, and this is the story in a brief summary. Peloton Therapeutics developed 3 compounds that have been broadly used. One is PT2385. This is the first compound that went into humans. Another one is PT2399. This is a tool compound that is very similar to PT2385 that we use for preclinical work. And the third one is PT2977, which is also known as Belzutifan. was renamed Belzutifan after Merck acquired Peloton Therapeutics. As you can see, these three compounds are all very similar. So the idea is that these compounds bind the HIF-2 alpha protein and will trigger the dissociation from HIF-1 beta, and this blocks transcription. And we tested these compounds in a library that we have of tumor graph or PDX models. And this is work also done by mentor Bill Kellen, where he also tested these compounds in xenograft mice. And essentially, in experiments that involved over 200 mice, we showed that treatment with this HIF-2 inhibitor, in this case referring to PT2399, suppressed the growth of kidney cancers in mice. These are patient clear cell renal cell carcinoma tumors that have been implanted in mice. And in these studies, there was greater activity with the HIF-2 inhibitor than there was with sunitinib, and this drug was also better tolerated, whereas mice treated with sunitinib lost weight, mice treated with the PT2399 did not. So we were very excited with this data. This data suggested that indeed the strategy was going to be working for kidney cancer patients. And again, as I mentioned, these experiments that we did in the laboratory involve over 250 mice. However, we found that resistance develops. So resistance to targeted therapies is a well-known problem. And the way we asked whether resistance was going to happen is by treating mice with for a very long period of time, as you can see here, for about 6 months. And as you can see, a tumor that was initially responsive then became resistant over time. I'm only showing you a little bit of data because I don't have much time in the presentation, but the HIF-2 inhibitor dissociates the HIF-2 complex, as can be seen here, but after the development of resistance, we found a reformation of the HIF-2 complex in these immunoprecipitation experiments. And then when we did sequencing, we identified in these two different mice. In one mouse, there was a mutation in HIF-2 alpha in the DNA not in the DNA, in the drug binding pocket. And we also found a mutation in HIF-1 beta, which was at the surface between HIF-2 alpha and HIF-1 beta, and this is represented here. So these raised concerns that maybe this would happen in patients, and I'm going to get back to that in a little bit. So there was a multi-center trial that maybe some of you in the audience participated in that was led from UT Southwestern by Kevin Corney. This was a phase 1 trial with PT2385. This is the first in human. This is the first in-class drug. And this drug showed that about 20% of the patients that had been heavily pretreated remained without progression on treatment for over a year. And that gave us a lot of hope. However, pharmacokinetic studies determined that there were a subset of patients that had very low levels in plasma of the drug. So there were a subset of patients that were metabolizing the drug quite quickly. And when investigators at Peloton correlated with the clinical data, they found, not surprisingly, that these patients that achieved low levels of the drug in circulation also did not benefit. So this was the reason why Peloton Therapeutics decided to invest in a second-generation inhibitor, which led to the development of Belzutifan, PT2977. Of course, it's called PT for Peloton Therapeutics. And essentially, they figured out how the drug was being metabolized, and they came up with a second-generation inhibitor that, as you can see here in monkeys, is a lot more stable than the first-generation inhibitor, and is also slightly more potent. In a companion study of the phase 1 trial, we showed that treatment with this drug resulted in dissociation of the complex. These are proximity ligation assays, and these are tumor biopsies, pre-treatment and on treatment. And if you look at these red dots, which represent the complexes, you can see that there is a diminution in the number of complexes. This is quantitated over here. And then if we did RNA sequencing, we could see that there was inhibition of the HIF-2 gene expression program in patient tumors. This was a patient of mine that participated in this phase 1 trial. He was treated with PT2385. He had been treated with 5 or 6 lines of therapy before then, and he did quite well. He was on treatment for a year without progression, but at the end of the year, he started to have some progression. You can see this renal mass, which is not another primary, but a metastasis beginning to fill up. And we did a biopsy of the renal mass, and we found by next-generation sequencing or conventional sequencing, in fact, the same mutation that we had predicted based on the studies in tumor grafts. So that goes to show that the tumor grafts have the ability not only to predict resistance, but also specifically the molecular mechanism. In progression biopsies, we could see that, unlike what I showed you earlier, there was no reduction in complex formation. And when we do gene expression analysis, at the time of progression, there was reactivation of the HIF2 program. So this shows that acquired resistance is a problem. We are seeing this in the clinic. And of course, this is likely to happen not just in a sporadic metastatic clear cell renal cell carcinoma, but also in VHL syndrome. And I think we may be hearing more about this from Marston and Samra. So in the context of a sporadic metastatic clear cell renal cell carcinoma, this is likely to be associated with mutations, as I have shown you. There may be other mechanisms. I presume, and this is my conjecture, that this is going to happen earlier than what we are going to see in VHL patients, and there is clinical data to support that, maybe more generalized in a sporadic and more likely to require changes in systemic therapy, whereas perhaps in the context of VHL syndrome, resistance may be more likely to be focal and could be managed with focal approaches. In the context of focal approaches, on my program, we've been interested in the development of stereotactic radiation for this purpose. And in fact, we conducted one of two prospective clinical trials, phase 2 trials, of stereotactic radiation for oligoprogressive disease. So these are patients that have metastatic kidney cancer that is progressing at a handful of sites. In these cases, one of the studies was fewer than 3 sites, the other one was fewer than 5 sites. And we showed that you can manage oligoprogression with stereotactic radiation with good local control rates, and you can delay a change in systemic therapy by the order of a year or so. So how are we going to manage acquired resistance? Well, there are second-generation drugs that could be effective, siRNA-based therapeutics, degraders, perhaps other approaches. Just going to show you a couple of slides from an siRNA that was developed by Arrowhead Pharmaceuticals. There is both a first generation, which I call here A1 HIF2, and a second generation, which I call A2 HIF2. And as you can see, these siRNAs are able to deplete not just wild-type HIF2 alpha, but also the resistant mutant HIF2 alpha. So we tested these siRNAs in our mouse platform. These are the mice that have been implanted with patient tumors. I'm just going to show you here one example, but this is published. Essentially, we found that these siRNAs were quite effective at blocking tumor growth. This is shown here. You can also see this here. And they were able to knock down HIF2 alpha in the human tumor implanted in the mouse. And the siRNA is being given intravenous once every 2 weeks. And this resulted in depletion of the protein, also by Western blot, and abrogation of the secretion of human VEGF. So these data led to a first phase 1 clinical trial, which I had the privilege to lead along with some of my colleagues at MD Anderson and elsewhere. And this trial, in my view, provides a proof of principle for siRNA therapy. So you can see this is actually a patient of mine who had a lot of progression just in the 2 weeks between treatments prior to starting on the trial, got 2 doses of the HIF2 inhibitor and only got 2 doses of the siRNA because she had a bleed and she had an acute coronary syndrome event, which precluded further drug administration. But as you can see, this subcarinal metastasis, which was progressing in 2 weeks, became and regressed, disqualified as a partial response. And this was the case despite the fact that there were only 2 doses of treatment for about 4 months. In tumor biopsies, we could see depletion of HIF2 alpha. And from this patient, we actually took one of the biopsies, and we implanted the tissue in mice, and we did a co-clinical trial, and we saw the same findings in the mice. We treated the mice implanted with that patient tumors with the siRNA, and as you can see here, we were able to see suppression of tumor growth. We were able to see depletion of HIF2 alpha, not only by immunohistochemistry, but also by Western blot. So far, I've shown you about the development of this first-in-class drug. I've shown you that acquired resistance develop. I've shown you a strategy to tackle acquired resistance with siRNA. There are others. But of course, another problem is innate resistance, right? Not everybody responds. And here what's interesting is that if we look at VHL patients, and this is data from Eric and Rama, most of the tumors do respond to the HIF2 inhibitor, but this is very different to what happens in the sporadic disease, where response rates, as we have seen in the registration trial, are in the order of 23 or so %, as opposed to 65% or so. So there is a fundamental difference between the responsiveness of clear cell renal cell carcinoma in VHL patients in the sporadic context. And we hypothesize, based on the data that we have obtained, that the sensitive tumors are characterized by higher levels of HIF-2 alpha than resistant tumors, which have lower levels of HIF-2 alpha and perhaps higher levels of HIF-1 alpha. This is quantitated here, and this is not the prettiest of Western blots, but I think it makes the point. So this led us to hypothesize that maybe HIF-2 alpha could serve as a biomarker. So I partnered with Shankar Sain, who is the director of our radiochemistry program, and what we did is we turned PT2385 into a molecular tracer. So we substituted a fluor atom for fluor 18. So this drug now becomes a PET tracer that we can use to visualize tumors that express HIF-2. So this is now shown here in mice, and I've run out of time, so I'm gonna go very quickly. So this is a mouse that has been implanted with a tumor that has low levels of HIF-2 alpha, as you can see here. There is very little nuclear stain, and a tumor on the other side that has high levels of HIF-2 alpha. The mouse has been injected with a PT2385 that has been labeled with F18, and you can see how the HIF-2 alpha expressing tumors lights up, whereas the other one doesn't. So this has led to an IND that we submitted to the FDA, and we have an ongoing clinical trial where we are studying this concept. So I'm going to finish off with the acknowledgements. So I'd like to thank the people in my team that did the work. We have here Sarania and Andrea and Vanina are managing the tumor graph platform. Eric Ma was very involved with siRNA experiments. Kevin Cornell led the phase 1 clinical trial. I work very closely with Payal Kapoor. Shankar San is the radiochemist that I work with. And of course, we worked with both the Peloton team and the siRNA team as well. And here is the funding. And would like to thank especially patients and their families. And if any of the students are interested in joining our team, send me an email. Thank you.
We have a few minutes for questions for Dr. Brugarolas. Please come up to the microphones.
Gianni.
Hey, Jim. Very nice talk. So very quick question. We're learning from prostate cancer, lung cancer, that a way to escape from targeted therapy is lineage plasticity or cell plasticity. Do you see similar phenomena in renal cancer when you shut down the HIF signaling?
Right. Thank you. So I think that's a very interesting question. This is something that has been seen, as you mentioned, a number of tumor types. That's sort of a change in lineage that results in resistance. We have not seen that thus far. And we are doing some work that suggests, interestingly, that HIF-2 may only be playing a role in the epithelial traditional clear cell renal cell carcinoma, but also at least in some sarcomatoid tumors. So the typical lineage plasticity that you might see from an epithelial to mesenchymal may not necessarily be associated with loss of HIF-2 dependency, since we believe that at least there is a subset of sarcomatoid tumors that are HIF-2 dependent.
Hi, I'm Claudio Casali da Roca from Brazil. Very nice talk.
Thank you.
In a very good review. I wonder if germline VHL mutation has something to do with HIF expression and also with Belzutifan response.
Right. Yeah. We think that VHL is inactivated in probably 75–80% of the sporadic clear cell renal cell carcinomas. And that's through mutation or methylation. Maybe the frequency is higher. And of course, you know that there can also be mutations in the longin component of the VHL complex. So I don't know whether that's what you were getting at or not, but it's unlikely, in my opinion, that the differences in response that we see in VHL patients in a sporadic disease are simply the result of a lower frequency of VHL inactivation in the sporadic context. Because if it were simply that, we would see response rates about 80% or so, because we think that 80% of the sporadic clear cell renal cell carcinoma have VHL inactivation. Does that answer your question? I don't know what's, okay, great.
Great talk. Thank you, Dr. Brugarolas. I was wondering, are we aware of any other proteins in the cell that Belzutifan might be able to target due to a similar structure or binding pocket?
Thank you. So one of the interesting things about Belzutifan is that it binds this very unusual cavity that I mentioned, which is present in the PAS-B domain of HIF-2 alpha. And in fact, in HIF-1 alpha, which is the most closely related protein, is much smaller. So it doesn't even target HIF-1 alpha. It's specific for HIF-2 alpha. And we showed that the drug does not dissociate HIF-1 complex, just the HIF-2 complex. So, and if you think about how the drug was developed to target this very unusual pocket, it may not be surprising that the drug is incredibly specific. And I can tell you that when you treat tumors that have very low levels of HIF-2 alpha, and you do RNA sequencing, which allows you to look at 20,000 variables, we see no changes in gene expression. If there were another target other than HIF-2, you would be seeing changes, right? But in cells that don't have HIF-2, you treat with Belzutifan, no changes. So this is, I would say, it rivals the specificity of antibodies, perhaps. It's a highly specific drug.
Karel Pacák, it was a very nice, beautiful lecture. I have a very quick question. You mentioned about the Belzutifan blocking interaction between HIF-1 beta and HIF-2 alpha. Do you think that this is only the mechanism how Belzutifan is working?
I think so. And the reason I say that is because, as I mentioned, we model resistance in the mice, and we also saw it in patients. And if you make a substitution of an amino acid within the cavity of HIF-2 alpha so that you fill the cavity with a larger side chain and Belzutifan cannot bind, you can restore tumor growth. So that goes to show that HIF-2 reactivation is sufficient to support tumor growth despite Belzutifan. So I think the evidence in that respect is very convincing. It also goes to show that HIF-2 is actually a core dependency, right, that the cell needs to mutate in order to be able to thrive.
Okay, thank you so much.
Sure. Rama?
Ram Srinivasan from the NCI. Jim, as usual, a fantastic talk. I always learn something new when you
Even though you've seen the slides many times.
My question relates to the clear emerging resistance to HIF-2 alpha inhibitors like Belzutifan.
Yeah.
And obviously the approach you and Nizar and others took to use an siRNA, which would potentially get around what seems at least like one potential mechanism for developing resistance, is very intriguing, right? And you showed some very nice preliminary data. Unfortunately, as we all know, the compound isn't being further developed because of neurotoxicity and other issues. What are your thoughts about exploring this further? Do you know if there's any effort afoot to reconstitute this approach? Because it seems very, very interesting and something that may actually be very, very useful in the field.
Absolutely. So what Rama is alluding to, which I didn't share with you for the sake of time, is that the siRNA nanoparticle had some neurotoxicity, and the company is not going forward. So I have approached several other companies that work on the siRNA field, including Alnylam, among others, to see whether they would be interested. And right now we have an in-house program where we have nanoparticles that we can load with siRNA. But to me, simply the notion that we can deliver siRNA into clear cell renal cell carcinoma in mice and humans, that opens a tremendous door, right? It's not just HIF-2 as a target, there could be any other target. And I also did not get into this, but the delivery is somewhat specific because you are targeting particular integrins that get expressed on the surface of the tumor. So we have a tumor-targeted and a tumor-directed delivery of siRNA with proof of principle in humans and also in preclinical models. And I think this is something that really deserves looking into because it could open a completely new field of treatment.
Definitely. Thank you.
Sure.
Jim, I will use the moderator's right, and I will violate for 1 minute. I have a question for you. So in your theory of resistance, there is a high HIF and there is a low HIF. Do the tumors with a low HIF have a different growth rate than the tumors with a high HIF? And is it the response and the resistance HIF dependent that both of the type of tumors? And what I'm alluding to is there any possibility that something else drives on the top of the HIF?
All right. So I'm going. This is how. Yes, thank you. You ask a number of different questions, and it's a challenging topic. So the idea is, and of course, this is particularly important in the context of a sporadic clear cell renal cell carcinoma. How we identify the patients that are likely to respond and those that are going to become resistant. I think one thing is clear, and that is if you don't have HIF-2 alpha, you are not going to respond. So I think we can all agree on that. I would say something else is also clear. There are some tumors, some clear cell renal cell carcinoma tumors that have very low levels of HIF-2 alpha. Okay. So at a minimum, one could argue that by looking at those tumors that have higher levels of HIF-2 alpha, we would be enriching for those tumors that are likely to be more responsive. So you asked several questions. One is, do we see a difference in the rate of growth of tumors that have HIF-2 alpha versus those that don't? We have not looked at that systematically, but I would say not something that has become apparent. As you know, HIF-1 has been suggested as a tumor suppressor. I'm not sure that it's always a tumor suppressor. I think in some contexts it could actually be an oncogene. It's interesting that when we treat with inhibitor, we tend to see an induction of HIF-1 alpha, as if the cell were trying to compensate by upregulating HIF-1 alpha, and there are clearly some tumors that express, and Celeste Simon and others have shown this, HIF-2 alpha and HIF-1 alpha, right? So, I really don't have a clear answer for you other than the things that I mentioned.